Good morning everyone. I welcome you all in the Department of Scientific Computing and Modeling and Simulation. It is my pleasure to introduce Dr. Ajay Limaye. Okay, so when I am introducing him, he is getting a bit tense, so I will briefly tell few things about him. Yeah. He was from the first batch of Bachelor of Computer Science course, what was BSc Computer Science then. So he was amongst the guinea pigs since, since those days, and because of these guinea pigs, your batch has got stabilized into graduation and post-graduation probably. So after doing his BCS, that time the post-graduation in computers was not available. So the only thing which these people could do was MA in mathematics. Okay, so Ajay had MA in mathematics and he started working with Professor Gadre in the domain of computational chemistry. Okay, and after working in that domain, it was not clear that where he would get admission for PhD with this mixed up background. But he continued to work and he is the first student of first PhD student of School of Scientific Computing. So he completed his PhD in 1997 and went to Australian National University at Canberra as postdoctoral fellow and then continued to work there as a research scientist for last 25 years. So amongst his many achievements, this Drishti, the software he will be talking about and the applications he would talk. But there are two achievements worth mentioning. In the Canberra centenary year, he was listed amongst top 100 scientists of Australia. And very recently, that is in 2022, he has received J.O. Westwood Medal of Royal Entomology Society. His software Drishti has wide-ranging applications, and without any buzzword, when the field was very new, a very consistent hard work has led to this development. So please look at it with open minds. Okay, I see many of the computer science students and MSc scientific computing students around. Please have your minds open, forget the buzzwords and real work. That is what I expect from this presentation today. I hand over to Ajay.
Thanks. Thank you, Smita, for these kind words. She's my senior. So, before going into Drishti, I would like to tell you the story about how Drishti came about to be. That was in 1989 when I passed out BCS. If I didn't know how to draw an exponential curve, you wouldn't have seen Drishti today. So there is a story behind it. In 1989, when I passed out the BCS course, we didn't have the MCS at that time. So, like most of the kids around, I was like told by really doing nothing. I did try applying for jobs. Well, you might say I did try. But what led me to the group led by Professor Gadre was the person named Chandrasekhar. He used to teach in Modern College. I did my BCS in Garware College. And somehow, Chandrasekhar was student of Professor Gadre. He did MSc Computer Science, I think. But I don't know, I don't remember the details, but I had met him and he told me that there is Professor Gadre at Department of Chemistry. Of course, I didn't know him at that time. He was looking for a couple of kids who could program. So I went to university. I came here to the university. I think that was the second time I had seen university. I went to the Department of Chemistry, searching for Professor Gadre. Didn't know how he looked. Went to his office. You know his office. Very down-to-earth person. And I inquired about the position, the project. He said, okay, give me an application. He gave me a sheet of paper, pencil, and that was my first application. It was 3-line application. Dear Professor Gadre, I would like to apply for this position. Thank you, Ajay Limaye. Just wrote something on the piece of paper. I gave him. He said, okay, come tomorrow for the interview. So I went for the interview. We had some students sitting there. I went for the interview. He knows everything. So Professor Gadre and Professor Saurabh Pal, who became director of NCL, so they were the two people who interviewed me. I don't remember many details of the interview that he took, but I remember one thing. He gave me the chalk. On the blackboard, he asked me to draw the curve for exponential. I didn't know the curve for the exponential, but I knew that exponential lied between 2 and 3. So I started, okay, 2 raised to 0 is 1, 2 raised to 1 is 2, 2 raised to 2 is 4, and so on. And then 2 raised to minus 1 and so on. So and then 3 is something like that, so I just drew the curve. He was very happy. I was probably one of the first students who did draw the curve. And then, I think before the selection, he had his youngest daughter sitting in the other room, and probably he said, he's not too dumb. So that's how this dumb fellow joined the group. And then within, I think, a year, I was upgraded from Mattha to Gadav. And I remained Gadav till the very end. So I joined in 1989, October, November or something like that, and I left this place in 1997. So you can imagine that we had a Sadi Sati. Who was Shani and who's Kundali, I don't know. You can guess. For me, it was a very good Sadi Sati. I had a beautiful time. I really enjoyed my time here at university. Myself and Professor Gadre and other students, we used to go on walks after 5 o'clock or 6 o'clock. And we used to discuss, we used to argue on various subjects. It was a wonderful time that I spent at this university. So if it were not for that exponential curve, you wouldn't have seen Drishti. So after my PhD, I joined the Australian National University Supercomputer Facility. So at that time, we had about 8 or 9 or 10 people in that group, the Australian National University Supercomputer Facility. This is in Canberra. At that time, we used to get all the funding from ANU, the Australian National University. But over the years, the funding model has changed, and now we have become NCI, National Computational Infrastructure. So we service all the major universities and research institutes in Australia. They are now our clients. So ANU is now one of our clients. We get all our funding from the federal government. So our supercomputer is all funded by the federal government, and all the major institutes, they funnel money for people like us. So we have got these two streams, and that's how we changed from ANU SF, which was purely ANU centric group, to now the national wide group. So we cater to all the researchers in Australia. Now, before going into Drishti, I'll talk about how I came about, how I started writing Drishti. So I joined as a computational scientist in ANUSF, and within about 8 or 9 months, I switched to visualization. I mean, I had always had interest in visualization, even during BCS. During those times in BCS, we used to be given choices, so we had electives. And in one of those, we had scientific visualization as the elective, but although unfortunately we couldn't take that elective because there was nobody to teach us. But I was always interested in visualization, and even during my PhD days, I had a fight with Professor Gadre to write the visualization software to visualize molecules. That was one of my PhD chapters. So using that as a leverage, I moved from scientific high performance computing to visualization. Now, what is visualization? Visualization is not just images. Visualization happens when you transform the data into insight using graphical means. That is visualization. So we use visualization, for example, Drishti or inner sight. So it is basically we are converting data into information. That is visualization. And the whole purpose of visualization is, one is communication and the other is to provide insights. No. I'll tell you. So communication and insight. It's not just pretty pictures. It is insight into the data. You have the data, you need to understand it, and visualization is the visual means. Now, why visualization? Because most of our brain, or significant part of our brain, is actually geared towards vision. So the way vision happens is the photons go through the eyes, then they go through the optic fiber, and then at the back you have a big visual cortex which processes all this information. And that's how you get the depth perception, color, and all these things. So major part of our brain is geared towards understanding visual information. And that's why there is a saying that an image says 1,000 words. It's because you can convey significant amount of information through a single picture or a movie or an image. So I was always interested in visualization, and luckily I got to start working in visualization in 1998. And my first project was with an art historian. We reconstructed the temple complex of Borobudur. Now Borobudur is situated in Yogyakarta Island, on the Yogyakarta Island in Indonesia. When the UNESCO when the researchers found that place is in ruins, UNESCO took over and it started rebuilding and has reconstructed the entire Borobudur. During that reconstruction phase, the French researchers had taken photographs of each and every bas relief of the temple. So that temple complex is, I think, 76 square meters. It's a huge one. It has got 5 levels, and at the top level there are 36 stupas with bodhisattvas. So we reconstructed the entire thing in virtual world. So at that time, that was in 1998, 99, we had something called as VRML. So there is HTML, there is also VRML, which is not used these days. So virtual reality modeling language. So I built the entire temple in that virtual reality modeling language, and then you could take the tours in that virtual world. So that was my first project. Then I also wrote the Python scripts or Python program to view that entire complex in 3D. So at that time we didn't have the 3D headsets. It used to be projected on a screen in front using two monitors, 2 projectors, and you used to wear what we call as active stereo. So you would get an image on the right eye and left eye. It would be synchronized with the projectors. And then I wrote the Python script so that you could, using the wand, you would be able to navigate and go through all the entire temporal complex. So that was my first project. Then I worked with a chemist, doing animations, calcium channel, potassium channel, and so on and so forth. And then I started to do mine. Before that, we also did a big project on a museum, but I'll come to that in the later part of my talk. So let me start with Drishti. How I started working with Drishti. In around 2001, 2002, the Applied Mathematics group at the Australian National University, they are not called Applied Maths, but they are called something like 3D, mesoscale physics and something like that. They are part of the Research School of Physics and Engineering. But these are amazing guys. They work from nanoscale to mesoscale. They work on a huge range of materials and levels of the sizes, right from nano to really big ones. So they were building microcity at that time. Now for those who don't know what is a microcity, it is very similar to city. City is computed tomography. The principle is that you have a sample in the center, then you have an X-ray emitter at one end, and the photographic plate or detector at the other end. So if you take an X-ray, it's just a photographic plate, so you get the X-ray where the bone is white and the other tissues are black and so on. The reason bone is white is because when you emit the X-rays, they pass through the body, they are absorbed by the bone because it is high density, it is essentially X-ray opaque material, and then some of the X-rays pass through the soft tissue and so on, and that's why you get white as the bone, and then more darker shades of gray for the softer tissues, which are more transparent to the X-rays. Same principle. What happens now is you have the sample in the center. What happens in medical city is that your X-ray transmitter and receiver, they move around the sample. Images are taken at each and every step, and then using radon transform, they are converted into a volume. So that's how you get a 3D volume. So what is a volume? Basically at every point, you have a function value, scalar, but you can also have vector value. So MRIs, you can get vector values and so on. What happens in microcity is you have the transmitter emitter and the sample is rotated. Now of course this is not possible with the human body. I mean you have a human body rotating like that. So you have the sample in the center which rotates and the images are taken. The other difference between microcity and medical CT is the resolution. With medical CT you get 0.5 millimeter resolution at most. The reason is you don't want to have high dosage because you would be then giving cancers or you will be killing the patient. With microcity, when the sample is live, of course the dosages are changed, but if you are say scanning rocks or fossils, you can have a very high dosage. Or if you have metals, then you might want to have a really high dosage because the X-rays then have to pass through. So depending upon how opaque it is to the X-rays, you change the source, the X-ray source, and the duration. In microcity, you can go for say hours scanning the sample. That way, you get a really high resolution, noise-free output for the volume. Whereas for the medical city, you just cannot afford. 1-2 minutes, that's it, and then the patient has to come out. So with microcity, you can get really beautiful high resolution images. Also with X-ray, what happens is because you are bombarding the sample with X-ray, the sample can get really hot. So people have scanned even ice cream, bread, chocolates, and all these materials in the city. The reason for scanning these materials is to understand the structure of these. For example, ice cream. I mean, you get all that texture is because of the porosity within the ice cream. Or tissue paper. I mean, tissue paper is mostly air. You get that fluffiness is because of the air. So basically, to understand the structure, we need to bombard with the X-rays. There is also optical tomography with the visible light, and they are used for example, in scanning zebrafish and so on, which are more transparent, and then they use the optical tomography there. So that's how I started writing Drishti. That they approached us to to see whether we can help them visualize the data that was going to come out from their microcity machine. At that time, there were programs that would do the job that they wanted, but looking at the quality of the images, I thought that I could do a better job, and I started writing the software, and that's how Drishti happened. And then over the years, I have got Drishti Pro, which is a touch screen. So on a touch screen, you can explore your data, and then also in the virtual reality. So what I plan to do is, I plan to go through the different applications that I've worked on over the years. No. No, just a moment. How do I move this slide? Oh. नाही माझी काय गडबड होते रे बघ रे काहीतरी चुकतंय माझं इकडे दिसतंय मला तिकडे दिसतंय दोन चालू आहेत का पॉवर पॉइंट्स Oh yeah, perfect. Okay, so we have, I've over the years I've worked with so many different researchers in variety of different disciplines. So I'll just briefly go through all these various disciplines. So one of them is genetics, epigenetics. So the group there at the Seattle Children's Hospital, they work on cleft palate. Now, 1 in 700 children are affected by cleft palate. So what happens is the palate doesn't form properly. So there is a gap between the palate. So I think there is so for example this one, that's the cleft palate. Now in order to understand why the cleft palate happens, they went through hundreds of chicken and mouse embryos. Now the reason they chose chicken and mouse embryos is because the genes that are responsible for the craniofacial development in humans is very similar to those in the chicken and mouse embryos. And that's why they chose those. Now how would you go on identifying which genes are affecting the craniofacial development? So in order to do that, you need to do cell labeling. So what is cell labeling? Cell labeling is essentially saying that that cell or those type of tissues are being affected or being affected because of these or this cluster of genes or this gene. So what they do is they basically tag with the fluorescent molecule which fluoresces under certain lighting conditions and then let the embryo grow through. So the tagging happens during the embryo stage and then they let the embryo grow through and then they scan. So for example this one, so this is a certain type of a gene that is expressed in these locations. So that's how they go through hundreds of such embryos and they actually go and manipulate these embryos by hand to change those genes, to modify those genes. So now once they have identified that these are the genes that are affecting craniofacial development, now they start modifying the gene so that you get that. You get the cleft palate. Now once you get the cleft palate, now you start identifying how to fix it. Medical research is cruel, it's brutal. So what they do, for example in here, they modify the gene such that only one part of the skull develops, whereas the other part of the skull stops development. So we have a symmetry. The reason symmetry happens is that the genes that are controlling the left and right part start and stop at almost the similar time. Now if you modify that, say for example stop this part and let this part grow, you will start getting curved, you will start getting bent. So that's what you are seeing here. For example, in this case, they let the teeth grow. So in the mouse, they let the teeth grow, then what happens to the skull? For example, here. So that's the normal type. That is called the wild type. Here, because that part is growing, the bottom part is growing, the upper part has no choice but to curl down. Same here. So you have got all these, basically all these modifications to see which genes affect what part and what happens to the body. Now once you identify that, you try to fix it. And what they have found is that with proper nutrition, you are able to fix cleft palate in the fetal stage. They have also worked on many different parts, for example, bone growth. What happens to the bone growth and so on. That's a CT scan of a chicken. Medical teaching is another area where Drishti is used. So what you are seeing here is the human skull. We took the skull from the cadaver, which was basically that part here, and then we micro CT, basically CT scanned it. And then there was an honor student who went through the scan and actually segmented each of these vessels. So we have a lot of vessels that go through this part of the body. It's a very vulnerable part of the body. Now, to come to that stage, what we first did was scan sheep neck. Now, what we found was, what for me it was new, and not for the medical guys. The muscles of the sheep, they are so strong compared to the humans. Now, the reason is, for the quadruped animals, their head is like that. So the head is all balanced because of the muscles here. Whereas for us, the head just lies on top of the shoulders. So we don't need to have really strong neck muscles. Whereas for the sheep, or for that matter dog or any, you need to have really strong muscles because your head is basically lying like that. And it needs to be like that. So that's why they have really strong muscles. And then they segment out the vessels. And that was used, that is being used for teaching. I've got a VR application for that. What the students do is they put the headset on. It's an Oculus headset. And when they put the headset on, they see the head in front of them on a pedestal. And then with a wand, they can point to any of these structures, and then the information of that structure, the vessel, would be floating in front of them. That was one part. The other part was you have the head in front of you, and then some vessel will light up, and then they have to come up with the right answer. So as a testing tutorial. So that's how that is the teaching material that they are using at the medical school in ANU. This one is another one. This is a fetal skull. Now, those who know a bit of medicine or anatomy would know that that part here is not fused. In adults, it is fused. So if you say, for example, touch baby's head, you will see, you could be seeing an indentation, or you can actually, it's a very soft part. Now the reason it is like that is because you need to allow the brain to grow. If the skull is fused at the birth, then there is no space for the brain to grow. So that's how it can tell us that it's a fetal skull. So this was done on Drishti Prayog, which is a touch table. And what these guys were studying was sinuses. So there are sinuses here, there are sinuses here, and here. So there are three basically groups of sinuses, and they drain out inside the skull. So if you want to drain out the sinuses, you do that, and it will drain out slowly, rather than that. So they studied the sinuses on the touch table. They could do digital dissection, so they could just slice open the digital data and they could see inside and they could identify the sinuses. These were again done by an honor student who used Drishti to create the entire display. This is a data from Visible Human Project. Now this person was a criminal who was put to death in 1991. After his death, his body was immersed in a blue gel and then cryogenically frozen, so minus 40 degrees. The body was frozen and then it was sliced at 3 millimeter resolution. And the photographs were taken at every slice. And then this is the reconstructed version of that using Drishti. The colors that you see are the colors in our body. That's what you see in this slice of something after cryogenically frozen. Now you're seeing the blue fringes. It's because the body was immersed in a blue dye, and I've basically removed that part. You see the blue fringes where the dye would be. So these are the sinuses. These are the eyeballs. These are another set of sinuses here. That's a blood vessel that's going like that here. Then these are the muscles. Lot of blood vessels that go through from here. So you can reconstruct the entire human. That's his leg. That's his foot. So these are the vessels you can see here. So that was the first visible human data set. There are now many visible human data sets. There are at least 5 to 6 Chinese human data sets. There are many Korean human data sets. I am not aware of any Indian human data set. So these are the muscles. Then there's some fat here. Fat is bluish, yellowish. Bones are also yellowish. And then you should see the muscles on the underneath the feet. They are really long, big muscles. That's why we can jump. So these are the muscles on the top. These are blood vessels, and these are the muscles at the bottom of our feet. They are really big. So again, this is used in teaching, or this is more for public engagement. So one important aspect of visualization is also public engagement. You need to engage the people. You need to communicate your ideas. And depending upon the audience, you change the communication style. So for example, if you want to communicate to people who are time poor, I would say politicians who are time poor. So in that case, you need to modify your talk or visualization so that you convey the idea instantly. So within 10 seconds, 15 seconds, your idea should be conveyed. But if you are talking or if you are presenting to a technical audience, then you have to modify your communication in such a way that you have that technical aspect. Otherwise, you are going to lose your audience, because they are not lay people. They understand the science behind it. They understand the phenomena behind it. So you have to target your communication in such a way that you engage your audience. This was with the plant genomics facility, the plant research facility. This was with St. Louis, but I have also worked with people at ANU. That's an orchid. That's from the ANU facility. These are the soil nodules in the bacterial colony in the soil nodules. So underneath the ground, the bacteria work with the plant in a symbiotic relationship. So these are the bacterial colonies of the soil. Then that's a maize root structure. Then they are also working on millets, the sorghum. Now, root growth is a very important topic. They want to understand how the roots grow in the soil. So what they do is, in case of maize at the Danforth facility, they have big sacks like that, and they have a granular medium. And then these plants are grown in the granular medium with the proper nutrients. And then every once in a while, they just drop the sac along with the granular material, and then they scan it. So they laser scan it. So sometimes they laser scan it, or sometimes they do the CT scan. So if it is a small one, then they do the CT scan. If it is a big one, then they just do the handheld laser scanner. They've also done experiments on carbon dioxide transmission from leaves down to the roots. So what they do is, they have the plant, then they enclose the plant in a carbon dioxide chamber. They do the PET scan of the entire plant, and they do it in for several times. So it's a time series. So you get a time series, and you can actually see the nutrients going from the root, from the leaves to the roots. So they do use all these technologies to understand how the plants grow. Then they also have big containers which are fully self-contained. They can control for lighting, they can control for moisture, they can control for soil, they can control for everything that they want. And then you can control a single parameter and then see how the plant grows. They have huge hundreds and thousands of tiny plants within that big chamber, and then they control it from outside, that I want to control just the lighting, or I want to control, say, day and night cycle, or certain frequency, or humidity, soil, nutrients, and then each of these parameters, they want to see each of these, how these parameters affect the growth of the plant. So for example, in Australia, because it is arid, they want to grow plants that can handle less water. Microfibers is also a big problem for sewerage. Now these days, most of the households use washing machines, and the fibers give off microfibers, and these are now then drained into sewerage system, and then they cling to each other and they become big balls. It's like microplastic. So that's also a huge problem for the sewerage facilities. I don't know what's in India, but in UNSW, New South Wales, they are working on this to understand how the fibers, what happens to the fibers with different wear and